What's up, guys? We'll go over how gametes, also known as sex cells, reproduce through the cool process of meiosis. The basics of meiosis is you start in one diploid cell, then you branch off into 4 haploid cells from the original diploid cell. Now, let's get into the phases of meiosis. First, meiosis starts here in interphase 1. In interphase 1, the chromosomes are replicated to form another copy of themselves. In this case, we started out with 3 chromosomes, and they replicate into 6 chromosomes. Then a nuclear membrane starts to form and the spindles start to replicate. After interphase comes prophase 1. In prophase 1, the nuclear membrane breaks down, that's a circle in the middle, and all of the chromosomes pair up into homologous pairs, giving us a total of 3 homologous pairs, or tetrads, of chromosomes. During the pair up, crossing over occurs where the chromosomes within the homologous pairs exchange some of their genetic information with each other, giving us unique chromosomes. From there, the 3 homologous pairs of chromosomes enter metaphase 1. The nuclear membrane is completely gone at this point, and the 3 homologous pairs are attached to the spindles. The spindles move the homologous pairs into the middle of the cell. Then we enter anaphase 1. The spindles pull apart the homologous pairs of chromosomes apart and moves them to opposite sides of the gamete. Then comes telophase 1 and cytokinesis. The cytoplasm gets divided in half inside the diploid cell during cytokinesis into 2 haploid cells. Each of these haploid cells has half of the original amount of chromosomes that the diploid cell had. And within the 2 sister cells, a nuclear membrane starts to form around the chromosomes. Then comes the second half of meiosis, called meiosis 2. The 2 sister cells enter prophase 2. Their nuclear membranes start to dissolve, and the spindles start replicating again within the 2 sister cells. No crossing over occurs in prophase 2. After prophase 2 comes metaphase 2. The nuclear membranes are gone, and the spindles attach themselves onto the chromosomes and pull them towards the middle. After that comes anaphase 2. The spindle fibers separate the centromeres. That's the part that attaches 2 sister chromatids together of each chromosome, which now gives us 6 chromatids in each sister cell. The spindle fibers move the chromatids to the opposite sides of the sister cells. Then comes the last and final step of meiosis, which is telophase 2 and cytokinesis. The 2 sister cells enter cytokinesis, so their cytoplasm gets divided in half, and now that gives us 4 gamete cells that are ready for reproduction. Now, what's the relationship between meiosis and Mendel's model of inheritance? Before we relate them together, what is Mendel's model of inheritance? Mendel's model shows that when you have 2 parents with different traits make an offspring, you can predict what traits will show up in their offsprings. There are 3 principles to Mendel's model of inheritance: the principle of segregation, independent assortment, and dominance. First off, the principle of segregation is that during reproduction, alleles from a parent are separated during the formation of gametes. This can be seen during meiosis, when the homologous pairs of chromosomes are separated during anaphase 1 and 2. Moving on, we have the principle of independent assortment, which claims that each pair of alleles segregate independently during the formation of gametes. It can be seen when chromosomes cross over during meiosis randomly, meaning that there is a random combination of alleles. And finally, the principle of dominance states that the dominant allele will show up in the offspring over the recessive allele. This relates to meiosis because the dominant allele is inside of male/female gamete during telophase 2, when the alleles are separated from each other and they are placed right inside gametes, and the dominant allele will show up in the offspring. That's it for this video. I hope you guys learned how meiosis works, and I'll see you guys later.